What?
In 2020, Jennifer Doudna won the Nobel Prize in Chemistry. But to understand how she got there, we gotta go back.
As a young girl growing up in Hawaii, exposure to diverse cultures and the natural beauty of islands helped inspire her curiosity. Her home life also played a major role in shaping her success. Her parents encouraged curiosity through reading, discussion, and debating. Their evenings were often spent together playing games, listening to music, and discussing ideas. Weekends were spent hiking in places like Hawaii Volcanoes National Park, which further deepened her interest in discovery. In the 6th grade, her father gave her a copy of James Watson's The Double Helix, and it changed everything for her. Captivated by the idea that the secrets of life were written in tiny molecules, just waiting to be discovered, she pursued chemistry at Pomona College before earning her PhD at Harvard University, where she worked alongside Jack Szostak studying catalytic RNA molecules, ribozymes, and their ability to self-replicate, offering clues about how early life on Earth may have emerged. After completing her doctorate, she moved to the University of Colorado Boulder for postdoctoral research with Nobel laureate Tom Cech, where she turned her attention to understanding the three-dimensional structure of RNA, considered a difficult and uncertain frontier at the time. Using X-ray crystallography, she helped reveal how RNA molecules fold into complex shapes that allow them to perform chemical reactions, much like proteins do. These discoveries established her as a leading figure in structural biology and RNA research, and laid the groundwork for everything that would follow. While Doudna was building her career in RNA research, a French microbiologist and geneticist named Emmanuel Charpentier had spent years studying Streptococcus pyogenes bacteria, where she made a key discovery, a small RNA molecule called tracer RNA that played a critical role in the CRISPR system. After which, Charpentier reached out to Doudna to collaborate. Together, they brought complementary expertise to a question researchers had long been trying to answer. Bacteria were clearly capable of recognizing and removing viral DNA from their own genomes, using the CRISPR-Cas9 system. But how exactly did this system work?
When they began working together, they already had several important pieces of the puzzle, pieces that had been accumulating for decades. The CRISPR sequences themselves had first been discovered accidentally in 1987 by Japanese scientist Yoshizumi Ishino, and it wasn't until 2005 to 2007 that researchers began to understand that these sequences were functioning as the bacterium's immune system. By the time Doudna and Charpentier began collaborating, the following had already been established. 1. Bacteria possess a genomic region called CRISPR, or clustered regularly interspaced short palindromic repeats, which functions as both a kind of immunological memory and a surveillance team. 2. When a virus attacks a bacterium, it injects its genetic material into the bacterial cell as part of its attempt to hijack the cell's machinery and replicate itself. 3. If the bacterium survives this infection, it doesn't simply forgive and forget. Instead, it captures short fragments of the viral DNA and stores these fragments within its own genome, between those repeating CRISPR sequences. These stored fragments of viral DNA are referred to as spacers. 4. This region of repeating CRISPR sequences interspersed with the viral spacers gets transcribed into RNA, which is then fragmented. Each of these fragments of CRISPR RNA contains one variable 5' end, which is made up of the approximately 20 nucleotides that match the viral target, and one constant 3' end, the repeated CRISPR sequence. And 5, they knew that a second RNA molecule called tracer RNA, whose role Charpentier had identified, and the enzyme Cas9, a protein that acts as molecular scissors capable of cutting double-stranded DNA, were both involved in the system. They could observe that somehow this complex was coming together and sifting through its own genome for foreign viral DNA, but they didn't understand yet how. Most importantly, they wanted to know, how did the CRISPR-Cas9 complex actually locate and recognize matching viral DNA sequences when a new infection occurred? Doudna and Charpentier collaborated for nearly 2 years, publishing a programmable dual RNA-guided DNA endonuclease in adaptive bacterial immunity on August 17, 2012. And what was in that paper was truly groundbreaking. The tracer RNA and CRISPR RNA interact directly, base pairing to form a stable duplex, and that duplex recruits Cas9, activating it. Now you have a 3-part complex that's ready to hunt. Cas9 scans along the DNA, looking for a short NGG sequence called a PAM, essentially a red flag. When the original copy of the viral DNA was added to the CRISPR array, they copied all but this short sequence, and its presence tells Cas9 that this is the real intruder. When Cas9 spots one, it stops, unwinds the double-stranded DNA, and lets the CRISPR RNA check against its copy of the viral DNA for a match. If the sequences align, Cas9 makes the cut. Once they understood the system, they realized that the only part actually dictating where Cas9 cuts is the variable approximately 20 nucleotide 5' end of the CRISPR RNA, the part that matches the viral sequence. Everything else, Cas9, PAM recognition, tracer RNA, is performing the same job each time, regardless of the target. In an attempt to simplify, they tried fusing the tracer RNA and CRISPR RNA into one continuous RNA molecule. And it worked the same, only now much easier to work with. Instead of 2 separate RNA molecules that have to find each other within a cell, they now had one continuous programmable molecule that could be aimed at any DNA sequence of their choosing. So you could say, swap out that 20 nucleotide sequence for one matching a disease-causing mutation, and boom, you've just handed the scissors a new set of instructions. Truly groundbreaking biological technology with an almost absurd number of applications, all wrapped up in a package so small it can't be seen with the naked eye.
In 2015, Doudna founded the Innovative Genomics Institute, pulling together a diverse group of scientists to research and develop genome editing tools to tackle a wide array of problems, from medical research issues to climate change. More recently, Doudna and the IGI are turning their attention to microbiomes. The trillions of microbes living in our bodies play a massive role in our health, and disruptions to those communities have been linked to diseases ranging from inflammatory bowel disease to Alzheimer's. Researchers at the IGI are now working to develop tools that can target problem-causing bacteria directly, intervening before they trigger chronic disease. But the human gut microbiomes aren't the only focus of IGI's research. Microbiomes in other animals are causing problems for the plant too. The bacteria living in the guts of cows produce methane as a byproduct. And at the scale of global livestock agriculture, that methane represents the single largest source of human-caused methane emissions. At the IGI, researchers are now working to develop tools to edit these microbial genomes directly, with the goal of reducing methane emissions and slowing climate change. As with any new groundbreaking advancements, though, this technology is not without controversy. Access to these treatments is still a major issue. Casgevy, the drug that treats sickle cell, currently costs $2.2 million per treatment. What's more, sickle cell disproportionately affects Black Americans, people who already experience less accessibility to healthcare, insurance, and medical representation. and people living in sub-Saharan Africa, the poorest region in the world.
There's also the controversy around the use of CRISPR-Cas9 in germ cells, editing parts of the genome that can be passed on to future generations. Unlike somatic cell editing, which only affects the individual being treated, germline edits are heritable. In 2018, Chinese researcher Ha Jiankui announced the birth of the world's first gene-edited babies, twin girls whose embryos he had edited using CRISPR-Cas9, forging ethical review documents and obscuring the true nature of the experiment from both the medical staff and the families involved. The scientific community responded with a unified condemnation, and Ha was sentenced to 3 years in prison. But those babies are still out there, living with edits to their genome that were never fully understood nor consented to. Sometimes the question with CRISPR isn't can we, but should we? And who gets to decide? Doudna's career has been defined by following curiosity wherever it leads, and the technology she helped create is still in its infancy. Diseases that have affected humanity for centuries are now closer than ever to a cure. The microbes contributing to disease and climate change may soon be turned into our allies. The molecular scissors are in our hands. And though there are some profound questions of ethics, access, and responsibility for us to navigate through, the future looks pretty remarkable.
I think that it really speaks to the fact that I think for many women, there's a feeling that no matter what they do, their work will never be recognized the way it would be if they were a man. And this recognition today, I think, just refutes that in a way, and it just honors the work that was started by a collaboration between two women, and who set out not to win the Nobel Prize, but to do really fundamental science that we thought was exciting and important and interesting. And if this prize changes any of that, I guess it sort of underscores that there's an importance to fundamental science that can really change. Fundamental science can change the world. I think this is a message that needs to get out there, because in many cases we hear the opposite. We hear that, why aren't scientists, why aren't we curing cancer? Why aren't we, working on very focused problems? And it's not that that isn't important, but the message here is that solutions to problems come from unexpected directions, and that's really the message of CRISPR. And I think it's a really important message to get out to young people, especially who are thinking about a career in science. And I always encourage students to pursue their passions, because we don't know where the next big discoveries and technologies are going to come from. And who knew that a bacterial immune system would in fact emerge as a world-changing technology for gene editing? But here we are.